Hello everyone, welcome to my channel Bindas Botany. Today we are going to discuss most important MCQ of plant tissue culture. Let's begin. Question number 1. Who is known as the father of tissue culture? Option A. Bonner. Option B. Leibach. Option C. Habertland. Option D. Gauthret. Correct answer is option C. Haberland. Then question number 2. An explant is option A. Dead plant. Option B. Part of the plant. Option C. Part of the plant used in tissue culture. Option D. None of the above. Correct answer is option C. Part of the plant used in tissue culture. The next question, question number 3. What is plant tissue culture? Option A. The technique of in vitro maintaining and growing cells. Option B. The technique of in vivo growing cells. Option C. The technique of growing plants in gardens. Option D. The technique of cutting plants. Correct answer is option A. The technique of in vitro maintaining and growing cells. The next question, question number 4. Which of the following tissues can be used as explant for regenerating complete plant through tissue culture? Options are options A. Shoot apical meristem. Option B. Embryo. Option C. Leaf segments. Option D. All of the above. Correct answer is option D. All of the above. And question number 5. Which of the following explants are suitable for the production of virus-free plants? Option A. Leaf segment. Option B. Seeds. Option C. Apical meristem. Option D. Stem cutting. Correct answer is option C. Apical meristem. Then question number 6. The process of combining the nuclear genomes of one parent with the cytoplasmic genome of the other parent is called as option A. Hybridization. Option B. Micropropagation. Option C. Regeneration. Option D. None of the above. Correct answer is option A. Hybridization. Then next question, question number 7. Which of the following component is not essential for murasage and skug media? Option A. Inorganic nutrients. Option B. Carbon source. Option C. Antibiotics. Option D. Organic nutrient. Correct answer is option C. Antibiotics. The next question, question number 8. Decrease in the pH of the media may result in option A. Increase in hardiness of the solidified medium. Option B. May interfere with the solubility of media salt. Option C. Interfere with the solidification of the medium and result in poor solidification. Option D. All of the above. Correct answer is option D. All of the above. The next question, question number 9. Somatic clonal variation can be present in which of the following plants? Option A. Plant regenerated through tissue culture. Option B. Plant generated through seeds. Option C. Plant generated through sexual reproduction, which includes fertilization of egg with pollen nuclei. Option D. None of the above. Correct answer is option A. Plant regenerated through tissue culture. Then question number 10. In vitro tissue culture can be used for the generation of option A. Virus-free plants. Option B. Somatic hybrid plants. Option C. Synthetic seeds. Option D. All of the above. Correct answer is option D. All of the above. Then question number 11. What is an explant? Option A. A part of a plant grown under soil. Option B. Any part of a plant taken out and grown in a test tube. Option C. A specific part of a plant grown in a test tube. Option D. Leaves grew under test tube. Correct answer is option B. Any part of a plant taken out and grown in a test tube. The next question, question number 12. What is callus? Option A. Tissues that grow to form an embryo. Option B. An unorganized actively dividing the mass of cells maintained in a culture. Option C. An insoluble carbohydrate. Option D. A tissue that grows from an embryo. Correct answer is G. Option B. An unorganized actively dividing the mass of cells maintained in a culture. The next question, question number 13. The growth of a plant tissue in artificial media is called option A. Gene expression, option B. Transgenesis, option C. Plant tissue culture, option D. Cell hybridization. Correct answer is G. Option C. Plant tissue culture. The next question, question number 14. The pair of hormones required for a callus to differentiate are option A. Ethylene and auxin, option B. Auxin and cytokinin, option C. Auxin and abscisic acid, option D. Cytokinin and gibberellin. Correct answer is option B. Auxin and cytokinin. When auxin concentration is high and cytokinin concentration is low, it helps for rooting, and when auxin concentration is low and cytokinin concentration is high, it helps for shooting. The next question, question number 15. The formation of embryology from the pollen grain in the tissue culture medium is due to option A. Organogenesis, option B. Test tube culture, option C. Double fertilization, option D. Cellular totipotency. Correct answer is option D. Cellular totipotency. The next question, question number 16. Which of the following is the main application of embryo culture? Option A. Clonal propagation. Option B. Production of embryos. Option C. Induction of somaclonal variations. Option D. Overcoming hybridization barriers. Correct answer is option D. Overcoming hybridization barriers. Then question number 17. In tissue culture of parenchyma, mitosis is accelerated in the presence of this. Option A. Auxin. Option B. Cytokinin. Option C. Gibberellin. Option D. Both auxin and cytokinin. Correct answer is option D. Both auxin and cytokinin. The next question, question number 18. Haploid plants can be obtained from option A. Anther culture. Option B. Bud culture. Option C. Leaf culture. Option D. Root culture. Correct answer is option A. Anther culture. It is a most important question. The next question, question number 19. In a plant tissue culture, the callus tissues are generated into a complete plantlet by altering the concentration of option A. Sugars, option B. Hormones, option C. Amino acids, option D. Vitamins and minerals. Correct answer is G. Option B. Hormones. The next question, question number 20. Which of the following vector is used in crop improvement and crop management? Option A. Agrobacterium, option B. Plasmid, option C. Cosmid, option D. Phasmid. The correct answer is J. Option A. Agrobacterium. Then question number 21. What is the dimethyl sulfoxide used for? Option A gelling agent, option B. Cryoprotectant, option C. Chelating agent, option D. An alkylating agent. Correct answer is G. Option B. Cryoprotectant. Then question number 22. The formation of embryology from the pollen grain in the tissue culture medium is due to option A. Organogenesis, option B. Test tube culture, option C. Double fertilization, option D. Cellular totipotency. Correct answer is option D. Cellular totipotency. The next question, question number 23. The production of secondary metabolites requires the use of this. Option A. Meristem, option B. Protoplast, option C. Auxiliary buds, option D. Cell suspension. Correct answer is G. Option D. Cell suspension. The next question, question number 24. Which type of plant is produced by plant tissue culture? Option A. Interspecific plant, option B. Dormant species, option C. Genetically modified species, option D. Hybrid species. Correct answer is option C. Genetically modified species. The next question, question number 25. Hybrids are produced by option A. The nucleus of one species but cytoplasm from both the parent species. Option B. The fusion of two same nuclei from the same species. Option C. The fusion of two different nuclei from different species. Option D. None of the above. Correct answer is option A. The nucleus of one species but cytoplasm from both the parent species. Then question number 26. Which of the following chemicals are most widely used for protoplast fusion? Option A. Mannitol, Option B, polyethylene glycol. Option C, sorbitol. Option D, manol. Correct answer is option B, polyethylene glycol or PEG. This is also a most important question. Then next question, question number 27. Which of the following plant cells show totipotency? Option A, cork cells. Option B, meristem. Option C, sieve tube. Option D, xylem vessel. Correct answer is option B, meristem. The next question, question number 28. Synthetic seeds are produced by the encapsulation of somatic embryos with option A, sodium acetate. Option B, sodium nitrate. Option C, sodium chloride. Option D, sodium alginate. Correct answer is option D, sodium alginate. Then question number 29. Which of the following medium is composed of chemically defined compounds? Option A, natural media. Option B, artificial media. Option C, synthetic media. Option D, none of the above. Correct answer is option C, synthetic media. Then next question, question number 30. What is callus? Option A, tissues that grow to form an embryo. Option B, an unorganized actively dividing the mass of cells maintained in a culture. Option C, an insoluble carbohydrate. Option D, a tissue that grows from an embryo. Correct answer is option B, an unorganized actively dividing the mass of cells maintained in a culture. The next question, question number 31. Agar is added to tissue culture for option A, providing micronutrient. Option B, encourage. Option C, stimulus for rooting. Option D, both A and B. Correct answer is option D, both A and B. Then question number 32. Parenchymatous cells that are made divide under controlled laboratory during tissue culture are option A, differentiated cells. Option B, dedifferentiated cells. Option C, undifferentiated cells. Option D, redifferentiated cells. Correct answer is option B, dedifferentiated cells. The next question, question number 33. Organogenesis or morphogenesis in tissue culture is controlled or regulated by phytohormones. The credit of this important discovery goes to option A, Skoog and Miller. Option B, Guha and Maheshwar. Option C, Calvin and Benson. Option D, Halperin and Wetherell. Correct answer is option A, Skoog and Miller. Then next question, question number 34. Concept of cellular totipotency was established by option A, White. Option B, Haberland. Option C, Stewart. Option D, Moore. Correct answer is option B, Haberland. Next question, question number 35. Cellular totipotency was first demonstrated by option A, F.C. Stewart. Option B, Robert Hooke. Option C, E. Swan. Option D, A. V. Leeuwenhoek. Correct answer is option A, F.C. Stewart. His full name is Frederick Champion Stewart. Then question number 36. A collection of all the allele of all the gene of a crop plant is called option A, germplasm collection. Option B, protoplasm collection. Option C, herbarium. Option D, none of the above. Correct answer is option A, germplasm collection. The next question, question number 37. Soma clones are option A, genetically identical to original plants. Option B, used to recover disease-free plant. Option C, somatic hybrids. Option D, sterile plants. Correct answer is option A, genetically identical to original plant. The next question, question number 38. Soma clonal variations can be obtained by option A, application of colchicine. Option B, irradiation with gamma rays. Option C, tissue culture. Option D, hybridization. Correct answer is option C, tissue culture. Then question number 39. Full form of MS medium is option A, Miller and Skoog. Option B, Murasage and Skoog. Option C, Michael and Smith. Option D, none of the above. Correct answer is option B, Murasage and Skoog. The next question, question number 40. Murasage and Skoog medium is commonly used for option A, bacterial culture. Option B, growing hydrophytes. Option C, separation of nucleic acids. Option D, tissue culture. Correct answer is option D, tissue culture. This question was asked in SSB PGT 2024. The next question number 41. In which of the following condition do the somaclonal variation appears? Option A, plant raised in tissue culture. Option B, plant exposed to gamma rays. Option C, plant growing in polluted soil or water. Option D, plant transferred by a recombinant gene technology. Correct answer is option A, plant raised in a tissue culture. The next question number 42. Sterilization of tissue culture medium is done by option A, autoclaving of medium at 121 degrees Celsius for 15 minutes. Option B, filtering the medium through fine sieve. Option C, mixing the medium with antifungal agents. Option D, keeping the medium at -20 degrees Celsius. Correct answer is option A, autoclaving of medium at 121 degrees Celsius for 15 minutes. The next question number 43. Tissue culture is option A, growth of specific plant structure on artificial medium. Option B, growth and multiplication of cells on artificial medium. Option C, cryogenic maintenance of tissues. Option D, maintenance, growth and differentiation of cells, tissues and organ on artificial medium. Correct answer is option D, maintenance, growth and differentiation of cell, tissues and organ on artificial medium. The next question number 44. Who among the following introduced tissue culture method of plant propagation? Option A, Normal Borlung. Option B, Gottlieb Haberland. Option C, E. K. Janaki-Ammal. Option D, M. S. Swaminathan. Correct answer is option B, Gottlieb Haberland. The next question number 45. In the preparation of culture media for tissue culture, DASH is used as a solidifying agent. Option A, carbohydrate. Option B, agar-agar. Option C, amino acids. Option D, growth hormone. Correct answer is option B, agar-agar. The next question number 46. Protoplast fusion causes option A, rapid growth of offspring. Option B, somatic hybridization. Option C, produce of useful allopolyploid. Option D, both B and C. Correct answer is option D, both B and C. The next question number 47. Agitation during tissue culture serves for option A, aeration of the tissue culture. Option B, constant mixing. Option C, breakage of cell aggregate in small particles. Option D, all of the above. The correct answer is option D, all of the above. Then question number 48. A major application of embryo culture is in option A, production of alkaloids. Option B, overcoming hybridization barriers. Option C, clonal propagation. Option D, induction of lethal mutations. The correct answer is option B, overcoming hybridization barriers. The next question number 49. Virus-free plant can be obtained through option A, grafting. Option B, callus culture. Option C, shoot tip culture. Option D, suspension culture. Correct answer is option C, shoot tip culture. Then question number 50. The enzyme used for separating the cells for protoplast culture are option A, cellulase and pectinase. Option B, protease and lipase. Option C, gyrase and helicase. Option D, endonuclease and ligase. The correct answer is option A, cellulase and pectinase. The cellule is made up of cellulose and pectin. Thus, the enzyme are required cellulase and pectinase. The next question number 51. In tissue culture media, the resource of the phytohormone is option A, glucose. Option B, agar-agar. Option C, coconut milk. Option D, micronutrient. Correct answer is option C, coconut milk. The next question number 52. The correct sequence in the tissue culture technique is option A, inoculation, then explant incubation, callus formation, then organogenesis, then preparation of medium, then transfer to field. Option B, preparation of medium, then inoculation, then explant incubation, then callus formation, then organogenesis, next transfer to field. Option C, preparation of medium, next inoculation, next explant incubation, next organogenesis, then callus formation, then transfer to field. Option D, explant incubation, then preparation of a medium, then inoculation, then organogenesis, then callus formation, then transfer to field. The correct answer is J. Option B, preparation of medium, then inoculation, then explant incubation, then callus formation, then organogenesis, then transfer to field. The next question, question number 53. Arrange the given step of the tissue culture process in the correct order. Option A, sterilization of culture media and explant. Option then 2, transfer of plant to culture medium. 3, selection of explant. 4, organogenesis in callus and formation of plantlets. 5, establishment of plantlet in the field. Options are 2, then 3, then 1, then 5, then 4. Option B, step 3, step 1, step 2, step 4, step 5. Then option C, step 5, then step 4, then step 3, then step 2, then step 1. Option D, step 2, then step 1, step 3, step 4, then step 5. Correct answer is option B, first step 3, then step 1, then step 2, then step 4, then step 5. Means first selection of a explant, then sterilization of culture media and explant, then transfer of explant to culture medium, then organogenesis in callus and formation of plantlets, then establishment of plantlet in the field. Next question, question number 54. Before inoculation into the culture media in tissue culture, the explant is sterilized by option A, autoclaving, option B, treatment with 1% sodium hypochlorite, option C, exposure to UV radiation, option D, boiling in water for 10 minutes. Correct answer is option B, treatment with 1% sodium hypochlorite. The next question, question number 55. Which chemical is used for surface sterilization of explant? Option A, sodium hypochlorite, option B, polyethylene glycol, option C, agar-agar, option D, ammonium nitrate. Correct answer is option A, sodium hypochlorite. The next question, question number 56 is totipotency refers to this. Option A, development of a fruit from flower in a culture. Option B, development of an organ from a cell in a culture medium. Option C, flowering in a culture medium. Option D, all of the above. Correct answer is option B, development of a organ from a cell in a culture medium. The next question, question number 57. The term totipotency refers to option A, cell to generate a whole plant. Option B, bud to generate a whole plant. Option C, seed germination. Option D, cell to enlarge in size. Correct answer is option A, cell to generate a whole plant. The capacity to generate a whole new plant from any cell is called totipotency. The next question, question number 58. This is the ability of a single cell to produce a fertile adult individual. Option A, pluripotency. Option B, multipotent. Option C, totipotency. Option D, all of the above. Correct answer is option C, totipotency. Then question number 59. Which of the following factor does not influence totipotency? Option A, relative humidity. Option B, non-reduced nitrogen. Option C, source of explant. Option D, light intensity. Correct answer is G, option B, non-reduced nitrogen. Then today's last question, question number 60. Essential requirement of an artificial medium in which explant is being regenerated is this. Option A, the medium should have a sulfur source. Option B, the medium should have a very low carbon concentration. Option C, the medium must provide a carbon source. Option D, the medium must provide a nitrogen donor. Correct answer is option C, the medium must provide a carbon source.
Thank you for watching this video. Please like, share, subscribe my channel and press the bell icon for notifications.